Okay, in this discussion, I want to talk about another difference between Mendel's laws or Mendel's rules, or what he even would have studied, versus something that's a little bit more complex, like it would take place in human beings. And that would be the concept of multiple alleles. Now, if you recall, when we talk about Mendelian genetics, anytime we talked about alleles for a particular trait, we were always discussing only two different alleles per trait. For example, a pea plant could have the allele big T for tall, and on its other chromosome from its other parent, it could have the little t for short. There are no more alleles other than the big T and the little t, tall and short. Well, in human beings and other organisms, it gets a little more complex than that. We can have what we call multiple alleles, or more than two choices for alleles. The example I'm going to use, and the one that is the most commonly taught, is the one of the ABO blood groups. Now, the way this is going to work, instead of just having two alleles, like big T and little t, I'm going to have the choice to have three alleles. We're going to call these big IA, big IB, and little I. And obviously A stands for type A blood, B stands for type B, but the I stands for type O blood. Now, just like with pea plants in their height, we have two alleles for our blood type. And the way you get these two alleles is obviously one from mom and one from dad. So if your dad were to give you type A blood and your mom were to give you type A blood, your blood type would be homozygous type A. And the reason why I said homozygous is because you are purebred type A. So the ways to get type A blood, other than being homozygous, so you can be homozygous, which is big I, big I, big IA, big IA, or you can be big IA and carry blood type O, which is little I. So both of these will give you the phenotype A, but obviously they're two totally different genotypes. For type B blood, you can be homozygous type B, which is big IB, or you can be heterozygous type B, which is big IB, little I. Both of these genotypes are for the phenotype B. To be type O, that's recessive, that's little I. And then to be type AB, you are big IA, big IB, and that gives you blood type AB. So these are all the different combinations, and what you need to be able to do is to be able to do a Punnett square, taking two different parents and figuring out what their child's blood types could possibly be based on the parent's genotype. So I'll go ahead and do an example of one of those for you. In this example, I'm going to say that the father is type A, but he's a carrier for O. Mom is type B, but she is a carrier for O. I chose this example on purpose because this is a very unique example, given that these two parents have the ability to have four children of different blood types. A 1 out of 4 chance of type AB, a 1 out of 4 chance of A carrier O, 1 out of 4 chance of B carrier O, and a 1 out of 4 chance of being type O. So then you need to be able to take all these different genotypes and come up with the probability for their children. Now, what are these type A and type B? Where does this stuff come from? If you recall, when we talked about cells, we talked about how your cells have markers that stick out. They're made of protein that stick out of the cell membrane. Those markers, I'm going to give those a name today. We're going to call those markers antigens. And so if I were to draw you a red blood cell, there, do you remember it's nice and round because it has to bounce off the other red blood cells. If I were to give you this red blood cell and put antigens sticking out, I'm going to label them type A. What this means is that this blood cell is type A, has the A antigen, but that also I can assume that this organism produces antibodies that will reject type B blood. So you never want to give somebody with type B blood to type A because it'll get chunky. It's called an agglutination when that gets chunky. So this person has type A blood and they have antibodies against B. This person is another red blood cell. Their antigens are type B, which means if you try to give them type A blood, they will reject it and it'll get chunky or agglutinate. If I want to, I can change both of these cells to type AB blood just by adding the other antigen to it as well. And these guys don't have antibodies against anything, and they can take anybody's blood. Type AB blood is known as the universal recipient. So what about type O blood? Well, in type O blood, they don't have antigens that mark either one of those, but they do produce antibodies that will reject both A and B. So therefore, these guys, they're known as being the universal donor, since they don't have those antigens that would get these cells to reject them. So type O, like I said, has no antigen sticking out.